Yeah, my name's Kinnari. I know it's really difficult, sorry. And I guess I'm one of the people that you don't envy because I'm going to try to talk about amino acid biosynthetic pathways. And amino acids are critical in that they are the constituents of the proteins which carry out the functions in the cell. And it's thought that the last universal common ancestor, or the root of the tree of life, is able to, was able to produce all currently available amino acids. So I'm interested in characterizing things at the last universal common ancestor, and obviously in the context of the progression of the central dogma or these other molecular processes. So, so the first question that I had with this project is whether the conservation of proteins associated with amino acid biosynthesis would be related to a characteristic of the amino acid, like the complexity of I have a bioinformatic approach where I compare features across species or the whole tree of life based on either the gene or protein level and extrapolate this information onto the last universal common ancestor. So a super cool postdoc in the lab, Aaron, who seems to be here in spirit as he got a shout out earlier too, curated this database called Lucapedia, which basically is a combination of several different data sets that attempt to characterize ancient characteristics of proteins based on these universal type distributions. And they use different approaches. So there's different types of ways you can approach this. Obviously, you can do it by gene families, by universally distributed protein motifs. Some of these studies use different structural perspectives, as in protein folds or groups of protein fold functions, and common reactions across the domains of life. So in terms of complexity, one of the references I'm using is proxy is the abundance in certain prebiotic synthesis experiments. So here I just have a table of the genetic code where I have the codon on the left and the corresponding amino acid on the right. And the shading just corresponds with higher abundance in these experiments. So I use a pretty simple bioinformatic pipeline. I start with a set of amino acid related proteins, and I match these with things that I found in at least 3 of the studies in the database, and I couple that with some ID matching, which I use the enzyme commission code for these proteins, which is just annotation for classes of enzyme function where you have 4 digits and each digit confers more specificity and description of the function of the enzyme. And I did this with and without the last digit, which usually describes some substrate specificity of the enzyme. So then I've mapped these things onto, specific pathways to see where things were conserved in biosynthesis. So here's one really cool pathway, and what you're looking at in red throughout the pathway are the 3-digit enzyme commission codes that are conserved or match at least 3 of these papers in the data set, in the database. And it looks a little daunting at first, but if I peel off things that are not conserved, you can see that there are these functional cores that seem to be conserved throughout the pathway. So while the main products in this pathway are serine, glycine, and threonine, you can still see some really close metabolic proximity, as in steps of reactions to get to other amino acids. There's tryptophan over here, cytosine, and methionine over here, and other amino acid biosynthetic pathways, as well as metabolism. So when I looked at the main products of this pathway in terms of these abundance proxies, there's no direct correlation between the conservation and the complexity or abundance of the amino acid, which was at first not what I expected. So another pathway that is probably really representative of the convergence and not entirely surprising is valine isoleucine and leucine pathway, in which the pathways are identical until you get to this last step of this last reaction. So that's probably the best example of convergent pathways given these sets. So throughout the data, So throughout the data, there were several nodal proteins or consistently represented enzymes in all of these pathways, or most of these pathways, and that was tryptophan synthase alpha chain and serine methylase. And so like I said, they're present in several of these super pathways, and they both confer different functions. Tryptophan synthase, obviously the last two steps of tryptophan biosynthesis, and serine methylase, catalyzing the serine to glycine reaction, as well as hydrolysis of tetrahydrofolate, which is just a common cofactor in amino acid metabolism as well as nucleotide metabolism. So I tried to find models of enzyme evolution that would possibly fit the data I have, and the Patchwork model seems to be a really popular model that may correspond, where you start out with these initially broadly reactive enzymes like red, green, and blue Pac-Man shape thing, and they do favor a certain reaction, but they're still broadly reactive. And after gene duplication, these things and selection from the environment, things become more specific in their function. So I thought maybe this could help explain, or maybe I'm seeing an artifact of this with these multifunctional nodes. Also, another popular theory is a semi-enzymatic theory, where you have reactions that become linked some way, and also incorporates the use of spontaneous reactions in development of these processes. So I guess my main conclusions are that you are able to identify certain functional cores that are conserved throughout these pathways, and possibly there's support for current models of enzyme evolution with these data. And I have to acknowledge my PI, Lauren Landweber, other members of the lab, two of which are in the audience but not in this picture, and of course Aaron Goldman, who's responsible for curating the database that I worked so closely with. So, thanks.
Have any questions for Kinnari?
Yeah.
Okay, well then I have one.
Sure.
I don't know a whole lot about where in the cells amino acid biosynthesis takes place, if it's all in one place or if it's spread out. Is there any locational tendencies? I realize it might not be a fair question, but
no, I'm not quite sure. I don't, and this is probably personal. I don't think of it as a localized set of reactions, but I'm not quite sure. Cool.
All right. Thanks. Kinnari.